What if I told you that scientists can now control a beating heart using nothing but light? Not surgery, not electricity, not drugs, just ordinary light. And this isn't some distant sci-fi dream. Researchers have just created heart muscle cells that respond to light like biological remote controls, opening the door to pacemakers that work with photons instead of painful electrical shocks. This could revolutionize how we treat heart disease forever. Welcome back to Mind and Marvel, where we explore the most incredible scientific breakthroughs reshaping our world. I'm your host, and today we're diving into research that sounds like pure magic but is grounded in cutting-edge bioengineering. Heart disease is the leading cause of death worldwide, affecting millions of people who rely on pacemakers, defibrillators, and other devices that use electrical shocks to keep their hearts beating properly. But what if we could control heart rhythm without any of the complications, pain, or limitations of electrical stimulation? Traditional pacemakers require invasive surgery, have limited battery life, and can cause complications. The electrical stimulation can be uncomfortable or even painful, and the devices need regular replacement. We show for the first time that light can be converted into cardiac stimulatory cues with synthetic materials made of biomolecules, said Herdeline Ann Ardona, Assistant Professor of Chemical and Biomolecular Engineering at UC Irvine. This breakthrough represents the first time scientists have created a new biomaterial that enables heart muscle cells to be stimulated by light without genetic modification or invasive methods. We're looking at a completely new approach to cardiac medicine that could make heart treatments safer, more precise, and far less invasive. But how exactly do you make heart cells respond to light? And what does this mean for the future of treating heart conditions? The science behind this breakthrough is absolutely fascinating, and it opens doors we never even knew existed. To understand this breakthrough, we first need to understand how heart cells normally work, and then how scientists have learned to make them respond to light. It's a journey that takes us into the fascinating world of optogenetics. Your heart beats because of electricity. Each heartbeat starts with electrical signals that travel through specialized heart muscle cells called cardiomyocytes. These cells receive electrical impulses and respond by contracting, creating the coordinated squeeze that pumps blood through your body. This electrical activity depends on tiny gates in the cell membrane called ion channels. When these channels open, electrically charged particles, ions like sodium, potassium, and calcium, flow in and out of the cell, creating the electrical signals that trigger contraction. Now, here's where it gets interesting. In nature, some organisms have evolved proteins that can respond directly to light. The most frequently used optogenetic protein is channel rhodopsin 2, CHR2, a light-gated, non-selective cation channel derived from green algae. Think of channel rhodopsin like a biological light switch. When light hits it, the protein changes shape and opens up, allowing ions to flow through. It's like having a door that automatically opens when you shine a flashlight on it. Traditional optogenetics involves genetic engineering. Scientists insert the genes for these light-sensitive proteins into cells, essentially reprogramming them to respond to light. Optical stimulation mediates the genetically expressed light-sensitive ion channels such as channel rhodopsin 2, CHR2, in the modified cells. This approach has shown incredible promise in neuroscience and has been used successfully in heart research. However, genetic modification comes with challenges: regulatory hurdles, potential immune reactions, and the complexity of getting modified genes into the right cells safely. This is where the UC Irvine breakthrough becomes revolutionary. Instead of genetically modifying the heart cells themselves, they've created synthetic biomaterials that can make normal heart cells light-sensitive without any genetic changes. The researchers developed synthetic materials made of biomolecules that can convert light into the kind of signals that heart cells naturally understand. It's like creating a universal translator that converts light language into heart language. This biomaterial approach offers several major advantages: no genetic modification required, potentially reversible effects, easier regulatory approval, and reduced risk of immune reactions. The heart cells remain genetically normal. They're just surrounded by materials that help them respond to light. In laboratory tests, when researchers shine light on heart cells treated with these biomaterials, the cells begin contracting in sync with the light pulses. It's like conducting an orchestra with a laser pointer instead of a baton. So how exactly do these synthetic biomaterials work? The mechanism is ingenious and involves some pretty sophisticated molecular engineering that's worth breaking down step by step. The biomaterials created by the UC Irvine team contain specially designed molecules that undergo physical or chemical changes when exposed to light. Think of it like molecular origami. The molecules literally change shape when light hits them. You might be familiar with this concept from photochromic sunglasses that darken in sunlight, or glow-in-the-dark materials. The principle is similar, but instead of just changing color, these biomaterials change in ways that affect nearby heart cells. When the light-sensitive molecules in the biomaterial change shape, they create physical changes in the local environment around heart cells. This can affect ion concentrations, create mechanical forces, or release chemical signals that the heart cells respond to. Remember, heart cells are exquisitely sensitive to their environment. Small changes in ion concentration, mechanical pressure, or chemical signals can trigger the ion channels in heart cell membranes to open or close, creating the electrical activity that makes cells contract. So the process works like this. Light hits the biomaterial, the light-sensitive molecules change conformation. This creates changes in the local environment. Heart cells detect these environmental changes through their normal sensing mechanisms, and the cells respond by contracting. What's brilliant about this approach is that it works with the heart cell's existing machinery. Instead of forcing cells to express foreign proteins, it manipulates their environment in ways they're already designed to respond to. The researchers can also tune these materials to respond to different wavelengths of light. This is crucial because different colors of light penetrate tissue to different depths. Red and near-infrared light can penetrate deeper into tissue than blue or green light. By engineering biomaterials that respond to longer wavelengths, researchers could potentially stimulate heart tissue deeper inside the body without needing invasive procedures to get light sources close to the heart. The materials are also designed to be biocompatible, meaning they don't cause harmful immune reactions or toxicity. They can potentially be applied temporarily and then naturally cleared from the body, unlike permanent genetic modifications. Another major advantage is temporal control. Unlike genetic modifications that are essentially permanent, these biomaterials can provide temporary light sensitivity, turn the light off, and the cells return to their normal behavior. This also offers unprecedented spatial precision. Researchers can illuminate specific areas of heart tissue with laser precision, potentially allowing them to stimulate exact regions of the heart that need intervention while leaving other areas unaffected. Now let's talk about why this breakthrough could completely revolutionize cardiac medicine. This can be beneficial for downstream medical applications, and the possibilities are staggering when you consider what this technology could replace and improve. First and most obviously, this could lead to completely new types of pacemakers. Instead of electrical leads that need to be surgically implanted directly into heart tissue, we could have optical pacemakers that stimulate the heart with light. Imagine a pacemaker that works through fiber optic cables instead of electrical wires, or even external light sources that can penetrate the chest wall to reach the heart. The reduction in invasiveness could be dramatic. Optical stimulation could eliminate many complications associated with electrical pacemakers. No risk of electrical interference from other devices, potentially much longer battery life since LEDs use very little power, and no painful electrical shocks during defibrillation. For treating dangerous heart rhythms, this technology could provide incredibly precise intervention. Opsins, such as channel rhodopsins, by conducting cations such as H+, Na+, K+, Ca2+, on light activation, can depolarize the cell membrane and hence initiate or prolong the cardiac action potential. Doctors could potentially stop dangerous arrhythmias by precisely stimulating specific areas of heart tissue with brief pulses of light, rather than delivering painful whole-heart electrical shocks. During cardiac surgery, surgeons could use light to temporarily pace the heart or test cardiac function without inserting electrodes. This could make procedures safer and give surgeons much more precise control during delicate operations. In research and drug development, this technology could revolutionize how we study heart function and test new cardiac medications. Scientists could create heart tissue models that respond precisely to light stimulation, allowing for much more controlled and reproducible experiments. For children with congenital heart defects, this could be particularly valuable. Growing children often need multiple pacemaker replacements as they outgrow their devices. Light-based systems could potentially be less invasive and more adaptable to growing bodies. There's even the possibility of remote cardiac control and monitoring. With the right technology, doctors could potentially adjust heart stimulation patterns remotely, or patients could have much more sophisticated home monitoring systems. This technology could also combine with other cutting-edge approaches. Imagine light-responsive biomaterials combined with 3D-printed heart patches, or integration with artificial intelligence systems that could automatically adjust light stimulation based on the patient's activity and needs. Beyond the heart, this approach could extend to other organs and medical applications. Light-controlled muscle stimulation for paralysis, precise neural stimulation for neurological conditions, or even controlled drug release systems activated by light. When you compare this to other emerging cardiac technologies, stem cell therapies, artificial hearts, gene therapies, light-based stimulation offers a unique combination of precision, safety, and reversibility that could complement or enhance these other approaches. Now, before we get too carried away with excitement, let's be realistic about where this technology stands today and what challenges need to be overcome before it reaches patients. Currently, this research has been demonstrated in laboratory-grown heart cells in culture dishes. That's a huge step, but it's still a long way from working in living human hearts with all their complexity. One of the biggest challenges is light delivery. Heart tissue is deep inside the body, surrounded by ribs, muscle, and other tissues that absorb and scatter light. Getting therapeutic levels of light to heart tissue without invasive procedures is a significant engineering challenge. While longer wavelengths of light can penetrate deeper into tissue, they also carry less energy per photon. The researchers need to find the right balance between penetration depth and activation efficiency. Safety testing will be extensive. Even though these biomaterials are designed to be biocompatible, they'll need to undergo rigorous testing to ensure they don't cause inflammation, immune reactions, or other side effects when used in living organisms. The typical pathway from laboratory discovery to clinical application usually takes 10-15 years. The researchers will need to demonstrate effectiveness in animal models before moving to human clinical trials. Regulatory approval will also be complex. This is a completely new type of medical device that doesn't fit neatly into existing regulatory categories. Agencies like the FDA will need to develop new frameworks for evaluating light-based cardiac therapies. Manufacturing these biomaterials consistently and at scale will require significant development. Each batch needs to have identical properties to ensure reliable and predictable therapeutic effects. There are also practical questions about how this technology would integrate with existing cardiac care. Cardiologists would need training in optical stimulation techniques, and hospitals would need new equipment and protocols. Cost will be another factor. While the technology could potentially reduce long-term healthcare costs by eliminating complications from electrical devices, the initial development and implementation costs could be substantial. Patient acceptance is also important. People are familiar with electrical pacemakers, but the concept of light-controlled heart devices might seem strange or concerning to some patients. The technology will also need to compete with other emerging cardiac therapies. Gene therapy, stem cell treatments, and advanced electrical devices are all evolving rapidly. However, the research is progressing rapidly. The fact that the initial proof of concept has been successful suggests that many of these challenges are solvable with continued development and investment. So, where does this groundbreaking research go from here? The UC Irvine team is already working on the next phases of development, and the implications are expanding beyond what they initially imagined. The immediate next step is testing these biomaterials in living animal models. This will help researchers understand how well the materials work in the complex environment of a living cardiovascular system, rather than simplified cell cultures. These studies will start with small animals and gradually progress to larger mammals with cardiovascular systems more similar to humans. Each step will provide crucial data about effectiveness, safety, and optimal delivery methods. Meanwhile, the engineers are working to optimize the biomaterials themselves. They're developing versions that respond to different wavelengths of light, last longer in biological environments, and provide more precise control over cellular responses. Equally important is developing better methods for delivering light to heart tissue. This might involve improved fiber optic systems, external light sources with better tissue penetration, or even implantable light sources powered wirelessly. The research team is also beginning collaborations with medical device companies that have the expertise and resources to develop commercial applications of this technology. Funding for continued research is coming from multiple sources, including government grants, private foundations, and potentially industry partnerships. This diverse funding base suggests strong confidence in the technology's potential. Other research groups are also exploring similar approaches, creating a healthy, competitive environment that should accelerate progress. Competition often drives innovation faster than isolated research efforts. The intellectual property landscape is also being developed, with patent applications filed to protect the core innovations while still allowing for collaborative research and development. Realistically, we're probably looking at 5-10 years before this technology might be ready for human clinical trials, and potentially 10-15 years before it could be widely available to patients. However, if the technology shows dramatic benefits in animal studies, there might be accelerated pathways for patients with life-threatening conditions who have exhausted other treatment options. While we've focused on cardiac applications, this breakthrough in light-controlled biomaterials has implications that extend far beyond heart medicine. We're potentially looking at a new paradigm for how we interface with and control biological systems. The same principles could be applied to neural stimulation for conditions like Parkinson's disease, depression, or epilepsy. Light-based neural stimulation could be more precise and less invasive than current electrical approaches. For people with paralysis or muscle weakness, light-activated muscle stimulation could provide new approaches to rehabilitation and functional restoration. There's also potential for light-activated drug delivery systems where medications are released precisely when and where they're needed in response to optical signals. In tissue engineering, light-responsive biomaterials could help create more sophisticated artificial organs and tissues that respond dynamically to their environment. This technology also aligns perfectly with the trend toward personalized medicine. Light-based therapies could be precisely tuned to individual patients' needs and adjusted in real time based on their response. Integration with wearable technology and Internet of Things health devices could create sophisticated networked healthcare systems where patients' conditions are monitored and treated automatically. Perhaps most importantly, this approach represents a shift toward more reversible, temporary medical interventions. Instead of permanent implants or irreversible procedures, patients could have treatments that can be adjusted, paused, or discontinued as their conditions change. On a global scale, if light-based therapies prove to be simpler and less expensive than current approaches, they could improve access to advanced cardiac care in developing regions where complex surgical procedures and device maintenance are challenging. So there you have it. A breakthrough that could fundamentally change how we treat heart disease and potentially many other medical conditions. Scientists at UC Irvine have shown for the first time that synthetic biomaterials can make heart cells light-sensitive without genetic modification. We've learned how this technology works by creating molecular switches that translate light signals into cellular responses, explored the revolutionary applications from painless pacemakers to precise arrhythmia treatment, and considered the challenges and timeline for bringing this to patients. While there's still significant development ahead, this research opens doors we never even knew existed. The possibility of controlling heart function with light represents a completely new paradigm in cardiac medicine. This work is continuing right now in laboratories around the world, with researchers pushing the boundaries of what's possible in biomedical engineering and optogenetics. If revolutionary discoveries like this fascinate you as much as they fascinate me, make sure to subscribe to Mind and Marvel and hit that notification bell. We're living in an incredible time for medical breakthroughs, with game-changing research emerging almost daily. I want to hear from you in the comments. What aspect of this light-controlled heart technology surprised you most? Do you or someone you know have heart conditions that could benefit from this type of innovation? What other medical applications can you imagine for light-responsive biomaterials? Remember, every heartbeat is a miracle of bioelectrical engineering, and now scientists are learning to enhance that miracle with the power of light. Thanks for joining me on this incredible journey into the future of cardiac medicine. Until next time, keep marveling at the amazing science that's reshaping our world.